Okay, good morning, everyone. Um, let's go ahead and start with our lecture today. I think one quick reminder is that hopefully all of you remembered to take your first quiz, which was due at 10:30 this morning. And so the quizzes, remember, it's up to you to take it when you're ready, but you have to remember to take it by class time. And then later this evening is when your first article summary assignment is due. So hopefully you've been well aware of that. And if you're not familiar with the instructions for that, go to La Lima, and in the resources folder, you will find examples of article summary assignments along with scoring rubric, just to let you know how they are scored, as well as the instructions. Okay. And so we will alternate every other week between the article summary assignment and the homework assignment, the short answer homework assignment that you turned in last Friday. So those will alternate week to week, but almost every week you still have a quiz to kind of keep you moving along. Okay? All right. Let me go ahead. Started. So today we're going to begin talking about this process of how normal healthy cells transform and start becoming cancer. And we're going to use specific examples looking at how certain viruses can be responsible for starting this process of healthy cells going rogue, right, going bad. So to start off with, let's talk about where the study of cancer has come from historically. So this is a painting of Hippocrates, the Greek physician who is often considered the grandfather of modern medicine. Right. So Hippocrates was the first sort of medical scientist, if you will, who wanted to go ahead and actually take ailments, the illnesses that he saw in his patients, and categorize them in ways that were objective and actually followed certain types of trails of evidence. Because remember, prior to this, because Hippocrates lived, around 460, 370 BC, a long time ago, right? Medical professionals, if you could even call them professionals, usually they're just considered wise men, right? They often treated diseases and illnesses as being supernatural in origin, right? It had to do with spirits and things like that. And in fact, to this day, a lot of the terminology that we use to describe different types of body components, things like that, still refer to a lot of these almost mythological kind of perceptions. Things like rheumatic diseases or humoral immunity all reference this ancient notion of things being the result of bad spirits or different types of spiritual processes happening within the body. So Hippocrates was the first to really want to say, no, there's nothing supernatural about this, right? These types of diseases are all based upon changes, underlying changes in the physiology of the body. And so that was the birth of modern medicine. That's why for medical students now, you take the Hippocratic Oath, right? That was all established by Hippocrates way back then. So cancer certainly has been around since the beginning of human time, recorded history. And you can certainly imagine that the appearance of cancer definitely was something that looked supernatural or the cause of bad spirits. Right? The name cancer is from Greek also, and it comes from carcinos, which means crab. That's why there is actually a very strong tie-in between the astrological symbol of Cancer, the crab, and the term cancer that we use to describe the disease. Why did they call it cancer? Because we imagine that early physicians like Hippocrates, when they would see things like solid tumors in patients, and they would actually examine them closer, maybe by actually dissecting the tissue and looking at it, what they saw in these solid tumors were these very messy things that looked like there were a lot of ill-defined boundaries to the tissue, oftentimes things that looked like these protrusions sticking out of them. In other words, almost like you had a crab underneath your skin, right? So that's the term cancer, and that's where it comes from. What about oncology, the study of cancer? It comes again from Greek, onkos, meaning swelling, right? So again, you can imagine oncology is the study of these mysterious swellings within the body, which we now know are tumors. So if you fast forward from 300 BC, zoom ahead up until like the early 1900s, okay, during that time, there was actually very little advancement done in understanding what cancer was. Right? But this story here, this guy, Peyton Rouse, was one of the most important sort of researchers because his breakthroughs in the early 1900s really helped shed a lot of light on the origins of where cancers can come from in our bodies. Peyton Rouse, he was a physician and a virologist at the University of Rochester in New York. And he loved studying viruses, and he loved studying the effects of viruses on cells. This is actually well before even we had a good understanding of what viruses really were. Right? One day, the story goes, right, Peyton Rouse was visited by a farmer, a chicken farmer, who was really concerned because this chicken farmer's prize chicken, the one that would win all these contests, right, was developing a tumor in its breast. And so he asked Peyton Rouse, can you help me cure my chicken? Right? And so Peyton Rouse was very excited about this. He said, sure. So he got the chicken, and Peyton Rouse went ahead and he killed the chicken to remove the tumor. Okay. So clearly he had his own sort of agenda here. Right. So in this case, it was a sarcoma, a solid tumor of muscle tissue. Again, remember, that's why it's called a sarcoma, muscle tissue, solid tumor. And what Peyton Rouse did was he broke it up into small chunks. He ground it up with sand to basically get the tissue as finely ground up as possible. And then he proceeded to filter that using water through a very fine pore filter, a filter that had holes that were so small that intact cells could not even get through. So basically what he was able to extract is whatever tiny bits of biological material could fit through this filter. What he did was he took this material and he injected it into a healthy chicken. And what he saw, to his amazement, right, was that the healthy chicken then developed a tumor, just like the original one. And so what Peyton Rouse concluded, and again, he's biased because he's a virologist, he said, whatever this thing is that can transmit the tumor from one chicken to another, it's smaller than an intact cell. So it's not a tumor cell itself, it's something even smaller than that. He concluded that it must be a virus. Right? And in this case, he was right. Right? So I think in another 20 years from then, other researchers actually confirmed the virus that Peyton Rouse was witnessing at work here. Right. So viruses can cause certain types of cancers. Not all viruses cause cancer. For example, let's be very clear, right? Just because you get the flu or a cold or something like that, right? Or the chicken pox, right? You're not going to be at elevated risk of developing cancer because of that. Certain types of viruses absolutely can, though, be linked to developing into certain types of cancers. Here is an example. What are some cancers that are linked to viruses? Cervical cancers in women, throat cancers in men are indisputably linked to this virus, human papillomavirus, or HPV. Right? We will examine that later. Different types of respiratory tract cancers, nasopharyngeal cancers, can be traced to Epstein-Barr virus, or EBV. Liver cancers can come from different types of viruses like hepatitis, B and C viruses, right? So these are the viruses, for example, that oftentimes in improperly processed seafoods, you might have the presence of functional virus particles in them. So you remember, like, what was it? 4 years ago, Genki Sushi had sort of this scare where people were developing hepatitis, right, from eating at Genki Sushi, and they traced it back to the scallops that they were serving, right? And I think they determined that there were different components. I don't remember right now what type of virus particle it was, or if it was bacterial in origin, right? But it's just another example of how different sort of pathogens can cause liver disease. And these pathogens, if they cause infection of the liver, hepatitis, that can in fact develop further into liver cancers. All right. Back then, 4 years ago or 5 years ago, I had just moved here. I'd never gone to Genki Sushi. And after that news, I was like, I'm never going to eat at Genki Sushi. But then it turns out these days I go there all the time with my kids, right? Because they like the little racing cars that deliver their California rolls to them. Okay, and then AIDS. AIDS is an example caused by human immunodeficiency virus, right? So AIDS itself, of course, is not a cancer, but AIDS is a syndrome whereby the person's immune system is so weakened and compromised that certain types of cancers that normally a healthy immune system can keep at bay, such as Kaposi sarcoma, become very prevalent in AIDS patients, and AIDS patients often die of these different types of cancers that in a roundabout way was caused by their initial infection to HIV. So around 20% of cancer types can be linked to viral infection. I want to be very clear on this so that you understand this. I'm not saying that 20% of all cancers are due to viruses, right? I'm not saying that, for example, if you look at all the people who are currently suffering from cancer, 20% of them, it's due to some virus infection. No, what I'm saying is if you were to make a list of all the different cancer types, you would find that about 1/5 of them, there is evidence linking them to some sort of viral infection. So as a result, viral infection contributes about 10 to 15% to the overall numbers of cancer cases that we have. So here are some cancer risk factors here. So for example, on the x-axis are just different examples of risk factors, and on the right is your relative contribution of that risk factor to developing cancer. So you can see by far the biggest risk are things like tobacco use, right? So things like tobacco use absolutely contributes tremendously to incidence of things like lung cancers and other types of tissue cancers like mouth cancers and things like that. Obesity is another big risk factor. It doesn't mean that obesity itself causes cancer. It just means that obesity carries with it a whole bunch of physiological changes in the body that put you at greater risk of developing cancer. And so here you can see pathogens, things like viruses, bacterial infections. So that's about 15%. Okay. These other types of factors you can see, like physical inactivity, diet, environmental exposures, right? These contribute in more modest amounts. But the idea is over a person's lifetime, the constant exposure to these things, like an unhealthy diet or occupational exposures like asbestos in the workplace, or things like lack of physical activity, over decades of life, right? These things are compounded and they increase the risk of cancer. But let's talk about viruses, because that's what we're talking about today. All right. So later, when scientists confirmed that Peyton Rouse's hypothesis that what he was seeing was a virus that was actually transmitting the cancer from one chicken to the other chicken. By the way, I don't know the end of the story. I don't know what happened when the farmer came back for his chicken. I don't know if Peyton Rouse just gave him another chicken or something like that. I should look that up because it's kind of unsatisfying to know. But anyhow, those scientists, once they confirmed that Peyton Rouse was right, they named this virus the Rouse sarcoma virus, right, or RSV. And that's what we see here. So this is a typical retrovirus, meaning that it consists of genomic material that is in the form of RNA. So in this case, it's a diploid, 2 molecules of RNA that code for all of the proteins that the virus needs to make new virus particles, right? It also has other proteins like integrases and reverse transcriptases that once this virus infects a host cell, these proteins are already pre-made and they go to work, helping the virus hijack the host cell. And then these are surrounded by an envelope or a capsule, right, which is a lipid bilayer with many different types of things embedded in it, including, most importantly, these proteins that are often referred to as spike proteins. Right? These are the proteins that are the right shape to bind to the surface of a host cell and allow the introduction of the viral particle into the host. Right? And so these are coded for by envelope genes. So you can see the viral genome then is really simple. If you were to look at one of these RNA molecules, you'd realize, yeah, it's just a set of very specific genes that each code for different proteins. So you have, for example, in this viral RNA genome here, you have these GAG proteins, which code for the core components, like the core membrane in yellow here. The Pol genes that code for things like reverse transcriptase and integrases. And then you have in green the envelope proteins that code for the various things that make up the outside, right? So that's a virus, right? A virus is a pretty simple thing compared to a living eukaryotic cell, like one of your or my body cells, right? But here's the question. Are viruses actually living things? Is something that simple actually considered living? Right? And I think this is oftentimes debatable. But my view, and I think the consensus view these days, is that no, viruses are not living things because they are really just some genetic material, some proteins surrounded by a capsule, right? It really can't do much by itself other than just be there, right? Its whole way of existing is just by chance encounters with other cells that this genetic material can get into and actually make that host cell produce other copies of the virus. Right? So you might say, well, then how come we often use the terms like when we're talking about, hand sanitizers and things? Oh, we got to kill the virus, right? Yeah, but that's just a general public way of thinking about things, right? We use kill in kind of like in general terms. I mean, you think about it, a virus cannot be alive, nor can it be really dead. It can just be disabled or destroyed, right? In the same way, we all oftentimes talk about our cell phones, right? You reach for your cell phone and you're like, oh, my cell phone died, right? It's not really dead. Where do viruses come from then? Right? And why is this so important in understanding how certain viruses can link to cancer? I use this analogy. I think some of you may have heard me use this analogy in other classes, but this was the class that I originally kind of introduced this. If we think of a normal cell as being filled with all kinds of wonderful proteins and other types of molecules that together make it a living, functional thing, right? We can think of that as kind of like a buffet, right? Yeah, I know. I always do these food analogies. A buffet has a wide assortment of different things to choose from, right? And let's say, for example, you are the head chef at a buffet at some really swanky high-end Waikiki hotel, right? And you're in charge of this buffet. You charge something like $50 per person, right, to go to this buffet. And you're in charge of making sure that the buffet always has all the components laid out in the proper way. And these components are presented, meaning that they're cut and fashioned and shaped and presented in the way that you expect them to be presented, right? So back in the kitchen, you have a lot of cooks and other short order kind of chefs that are preparing these things, right? So you need to teach them, for example, yes, for this buffet, when you get the cheese, right, you need to cut the cheese into cubes. And that's how you can set them out into this little cube tray, a tray of cheese. Or the bread, you also do in chunks like this here, because this is how it works in order to have this famous Waikiki buffet, maintain its standards. Okay. So the people back in the kitchen, they're doing this. They follow those instructions. But every so often, they kind of screw up, right? Maybe one of them misunderstood what you told them. Maybe one of them is new. So instead of breaking the bread into chunks, they slice the bread into slices, right? And you say, no, that's wrong. Throw that away, right? That's not right. We cannot use that for our buffet. if that then happens over time and enough accidents happen over time, what might eventually happen is one day the people in the kitchen realize, oh, we can take these mistakes, the bread that's sliced wrong, the cheese that's sliced wrong, right? And put it together into something new. So if a normal cell is like the buffet, a virus is like a sandwich that's made of components from the buffet that have been altered in a way that now they function to create something different. Importantly, the ingredients for the sandwich still depend upon the buffet. Right? You cannot have the sandwich go off and make other sandwiches by itself. It still has to be able to draw things from the buffet. But the point is, maybe the cooks in the kitchen realize, hey, you know what? People, the fancy tourists are coming in and spending $50 for the buffet. We can sell these sandwiches out the back door for like $5 a pop, right? And suddenly these sandwiches become super popular, right? But unfortunately, the day that the buffet closes down, there's no more sandwiches. So that's what I'm talking about here, right? Viruses are like little glitches that happen during evolution where certain cell components got usurped or borrowed, if you want to think about that way, and the right combination of little mistakes happened at the right time so that you could actually have the creation of a virus particle that has genetic material, proteins, a lipid capsule. And you say, that's hard to imagine, right? How could that happen in a string of accidents? Well, we're not talking about something that happened over 5, 10, 20, or 100 years, right? We are talking about something that has had hundreds of millions of years to happen, right? So these glitches, these viruses, are little mistakes that are carried along with our species, right? So, of course, in a virus, it's not enough just to say, I'm going to use some bread, some ham, and some cheese and make a sandwich, right? What we're really talking about in terms of glitches are the mistakes that are happening in the genetic material that code for the shape of these proteins, right? Just like the cooks in the kitchen, once they decide, I think I'm just going to make sandwiches instead of, helping to support the buffet, that knowledge of how to cut the bread into the right shape, the cheese and stuff, that's what's being transmitted, right? So many individual mistakes or mutations in prepping the buffet ingredients, they end up getting discarded by natural selection, right? Because the head chef goes in and yells at the other chefs, throw that away, we don't need that. But over time, if the right combination of mistakes come together, you can have something new, right? And it potentially can be preferentially selected for. Okay, so what does that have to do with our discussion of transformation of cells, right? The term transformation has to do with when you have a normal healthy cell and it starts down the pathway of disobeying the normal healthy rules that govern its cell biology, and it starts going rogue, starts going bad, right? We say that process is transformation. So in cancer speak, transformation has a very specific meaning. Right? Here we have an example of some chicken fibroblast cells. So these are chicken, connective tissue cells that are grown in a dish. And what these researchers have done is they've gone ahead and essentially sprinkled RSV viral particles, Rous sarcoma virus particles, into this dish and mixed it around in the media. And what you see over time is that the morphology of these healthy chicken fibroblast cells begin to change. On the right here is what the normal fibroblasts look like, right? This is their healthy, normal, differentiated morphology, right? As they serve to be connective tissue inside of the chicken. These cells in the middle, you can see something clearly is happening here. The hallmark is what? Instead of having that nice, long, spindly shape of the normal cells, they're now becoming just these balls, right? So what does that mean? They are de-differentiating, right? They are reverting in some ways back to more like an earlier stem cell-like shape, right? That's a key evidence here. So what is going on in these cells because of this virus to cause the transformation? If we look at them in different ways, here's a side view of that same dish. It's important to keep in mind that these fibroblasts grown in culture typically behave very well. They grow as a single cell monolayer, right? That's why if you look at this image over here in the middle, they kind of look like little cobblestones, right? They very nicely grow until they bump into a neighboring fibroblast, and then they kind of stop, and they're very happy then, just taking up their own little space. And you get this nice little cobblestone pattern. Okay? Here's another view. If you look it down, here are some squamous cells here, right? So these are flat squamous cells, right? These top ones are more like the fibroblasts from the chicken. These are squamous epithelium, nice and flat. But you can see, right, they respect each other's boundaries, right? Where they end up touching each other, they stop and they form a monolayer. These cells in culture then, when they are infected with RSV particles, they begin to transform. They begin to ball up and lose their normal differentiated shape. And importantly, they get kind of rowdy, right? They start piling up on each other. All right, so here you can see these transformed cells are beginning to pile up in what we call little focal points on the dish, or foci, right? These foci over time can comprise thousands and thousands of cells, right, to the point where you can actually see on the dish, right, even without a microscope, little specks. And each of these little specks is a group of transformed cells. The epithelial cells below, you can see they are transforming as well. From their nice flat squamous morphology, they are also balling up, right, as they lose their normal differentiation. So loss of contact inhibition, meaning once they're bumping into each other, they no longer care. Right? Contact inhibition means normally when they bump into each other, they stop growing. Right? The transformed cells lose that contact inhibition, and they lose their normal differentiation. Here is another important thing. I said that certain types of epithelial cells and those fibroblasts, when you grow them in culture, they will grow on a nice surface layer, right? They will be on the surface of the plasticware. They won't be floating around. Okay. That's very much because things like epithelial cells, as I've already introduced to you, right, their normal function is to behave well and stay attached to a surface. For example, the basement membrane and the stroma of our connective tissues in our body. Right? When they come loose from that, normally they're not happy about that and they'll stop growing. Okay. But in transformed cells, what you see happening is as they pile up, not only do they pile up, but they can actually then lift up off of the dish and now be free floating in the media as these clumps of cells that continue to grow. So they become anchorage independent. Right? And I think we got to this before in our introduction, but you can already imagine if you have tumor cells that lose their anchorage independent growth and now are able to continue growing once they are released and are passing through the blood, that's bad news, right, for in terms of the spread of things like cancer. So what role then does the genome of the virus play in actually not only starting transformation, but sustaining transformation? Because after all, transformation is not an overnight process, right? It's not like one day the cell is healthy and normal, and then a virus particle comes in, and then boom, the next day it's this transformed cell. No, it's a gradual process. That means not only does the virus genome that has infected the cell started the transformation process somehow, but it is sustained, right? What role does the virus genome play in that? Here is a wonderful classic early example that really demonstrated that the influence of the viral genome needed to still be at work inside of a transforming cell in order for that cell to continue going rogue and getting worse and worse and worse down that pathway of transformation. Right? So this was an experiment done, I think, in the 60s at UC Berkeley, my alma mater. And what these researchers wanted to ask the question of is it just enough to infect cells with a viral genome, right? Or does that viral genome still have to somehow be there in those cells the whole time that the cell is transforming? What they did was they took some, sorry, viruses. I think this was Rous sarcoma virus that they did, and they selected for mutations in this virus so that certain proteins of the virus were mutated. They would only work at 37. They would only work at a elevated temperature. So normally, the proteins inside of the Rous sarcoma virus, once they are expressed by the host cell, I mean, it's in a body that's 37 degrees Celsius, right? In a homeothermic tissue environment. So it makes sense that the proteins are designed to function. They are the right shape to function at 37 degrees. And if you were to warm things up, right, to let's say 40 degrees or higher, then these proteins might lose some of their normal native three-dimensional shape, and therefore the virus might not be able to function properly. Okay. So what these UC Berkeley researchers did was they on purpose tried to grow RSV particles in cells at elevated temperatures. And by doing so, they selected for certain mutations in the virus that allowed then virus particles to function at a higher temperature. Okay. Those mutant forms of the virus, once you tried to grow them at the lower temperature, ironically, they were now messed up, right? Because they had been selected for to optimal grow at higher temperatures. So you could not grow them at lower temperatures. So that's what they used to do this experiment. They took some normal healthy host cells, maybe chicken fibroblasts, maybe human cells, for example, and infected it with these normal virus proteins, right? And you can see that, wait, what did I do here? Oh, no, I'm sorry. I think I messed up the story. It's that they actually selected for virus particles that would only work at 37 degrees Celsius, not that they would work at 41 degrees. Okay. And so they infected these cells with these virus particles, keeping the cells at 37 degrees. And sure enough, when they infected them with this RSV mutant, right, that would function properly at 37 degrees, right, the cells they saw would transform. They would start changing their shape. Okay. But because this was a mutant form of the RSV that would only work at 37, okay, they could then raise the temperature of their cell cultures to 41. And at 41 degrees, then the proteins inside of the RSV particle, right, would not be folded properly, and the virus particle would stop kind of functioning correctly. And what they saw was that if you did this early enough, just maybe a few days into the process here, right, suddenly these transformed cells would go back to looking normal. Okay. So in other words, by removing the influence of these viral genomes inside of these infected cells, they could reverse the transformation if they did it early enough. And sure enough, to play with that a little bit more, if they then lowered the temperature back down again, okay, they saw that these cells would once again transform. Okay? If you wait too long, so I think in this is, just a few days from the start of this experiment, you could go back and forth and actually make these cells revert back to normal if you were able to somehow prevent the viral genome from having an influence in them. But if you wait too long and allow these cells to transform too far, then you can no longer do this. Okay. But this was early evidence that whatever was happening involves the presence of the viral genome being sustained in these cells in order to encourage transformation to continue. All right. Here are some examples of different virus families and the types of genomes that they have in terms of their size. I don't think any of you are surprised to know that a viral genome is way smaller than, let's say, a human genome, right? But maybe some of you don't have a good sense of how small some of these viral genomes really are. So here are examples of some DNA viruses. So in other words, the genetic material that they carry around is in the form of DNA. And here's some example of RNA viruses like we've been talking about thus far. So things like human papillomavirus, HPV, right, is a DNA virus. It belongs to the Papova family. The size of its genome is around 8 kilobases. So about 8,000 base pairs of DNA is the entire genome of this. So you say 8,000, is that big? Is that small? Remember, a human genome has something on the order of 3.2 billion base pairs, right? So 8,000 base pairs is tiny. So it really makes you realize that all the genetic information required to make a virus particle can be contained in a relatively tiny bit of nucleic acid. Things like the Rous sarcoma virus, it's about the same size, about 9 kilobases. Right? So things like these virus families are named. They have kind of funny names like Papova, adenoviruses, pox viruses, right? Things like the Papova viruses are named because they form papillomas, right? So papillomas are these solid tumors. I think papillo is the same sort of prefix, meaning like, the papilla, little bumps like in your tongue or on your the lining of your intestines. In this case, they form papillomas, they form polyomas, and there's the presence of vacuoles that appear inside of infected cells. So that's why these are called papovaviruses. These are attractive for cancer researchers because of their relatively small genome size. Because you're talking about only 8 kb, you can very easily appreciate the entire map of the papovavirus's genome, right? And appreciate that all the genetic information that codes for how this virus particle is made, right, is there in front of you. The SV40 virus, right, is a good example of a papovavirus. So SV40 is named this way because it is a monkey virus. So it was originally called simian virus, so monkey virus type 40. And it's a good example of a papovavirus, as that is very well understood in cancer research. So here is an electron micrograph of the capsule of an SV40. All right, so you can see here, it's very obvious these are the protein spikes on its surface that are the right shape, the surfaces on them, so that they can interact with different features of a host cell, like a monkey cell, and that's how they stick and gain entry. Here you can also see these are monkey cells that are infected by SV40, and sure enough, you can see inside of these are these vacuoles, right? So that's all part of why they're called papovaviruses. So here is the SV40 genome. So these are the complete genomes of SV40 particles that have been broken up. Right? So in this case, an SV40 genome is about 5 kilobases, very small. So again, very attractive for cancer research. Right? If you look at this, it's kind of amazing if you think about it, right? So this little ring of DNA here that is 5,000 base pairs, it's amazing to look at this and realize that something in here, sequences within this chain, are enough to cause normal healthy cells to start transforming into cancer, right? And so what are these? Next time, we will go into an explanation of what these different elements are that actually cause the transformation, right? So when Rous identified the Rous sarcoma virus, and this became a model for other researchers to try to understand what genetic components of virus particles actually cause transformation, the next step was to really understand mechanically, mechanistically, what is happening inside of host cells that have this viral DNA or viral genome to cause the transformation to happen. So we will talk about that next time. But to kind of wrap up today's discussion, this is sort of a summary, right, of properties that are associated with transformed cells. So these are things that we've talked about, but now in the context of actually understanding cells that are transforming, I want you to think about this, right? So we discussed altered morphology, so rounded shape, right? They appear a certain way under the microscope. Today, we looked at how you can see loss of contact inhibition. So they gain the ability to grow over each other. So in cultures, these would form little piles of cells or foci. In living tissues, these would be the start of things like little tumors. Right? Ability to grow without attachment to solid substrates. So we call this anchorage independent growth. Right? And so we saw examples of cells that can form little balls of growing cells that are floating around in things like media. Right? And of course, in our bodies, that would be little groups of cells coming off of the tumor and being able to continue growing, floating around in the blood. Ability to proliferate indefinitely. This concept of immortal cells, I think, is a very key concept in terms of understanding cancer. And we will discuss this whole idea of immortalization some other time. Other things that we didn't specifically talk about today, but are also important to consider, is that as cells transform and they become further and further away from their normal physiological cell behavior, right, in order for them to continue growing under conditions where they shouldn't be growing, means by definition that they have reduced dependence or reduced requirements on actually listening to other messages coming from tissues that normally would be telling them to divide or to stop dividing, right? So reduced requirements for mitogenic growth factors is a hallmark of transforming cells. High saturation density, right? I mean, that goes hand in hand with proliferation. It just means that normally healthy cells, they would hate it if they were all piled up together, right? They would stop growing and many of them might just die. But transformed cells, right, they can continue growing even though they're really packed in there. Inability to halt proliferation in response to taking away different types of growth factors. That's kind of similar to this point about reduced requirement for mitogenic growth factors, but we'll see how these 2 points are slightly different and how they, when we discuss them in more depth, help us illustrate slightly different concepts. Increased transport of glucose. I mentioned this in a previous lecture, that certain mutations in cancer cells that they stumble upon will allow them different metabolic growth advantages. So for example, certain mutations that end up resulting in increased transport of glucose into the cells so that the cells can increase their ability to do things like glycolysis to get energy, the much needed energy in the form of ATP from them. And of course, tumorigenicity. I mean, tumorigenicity, it's kind of a funny property to describe here, but really it kind of wraps up all of these things together. And it just means a hallmark of transforming cells is that, yes, right, they can lead to the formation of tumors. Okay. Okay, so let's go ahead and stop there. Let me stop the recording.